Greetings and welcome back. So this is a relatively short but hopefully fun little exercise. So what we're going to do is basically what I like to call the biological numeracy exam. It's an exam, but it's not graded. You're the one grading it yourself. And what I want you to do is please grab a piece of paper or your iPad and open up Notability, however you do it. And I think there's on the order of 15 questions. I'm just going to go through and read each one of them and write down your answers. And then in the next vignette, what we'll do is we'll spend a significant amount of time trying to reason out each of the different answers. And in some ways, this was inspired by what's called Gapminder. So the Gapminder exam is something I would highly encourage you to go do even before you do this one right now. So if you just go type Gapminder test, you'll be brought to a page. It's updated relative to the one that I show you here, and just as an interesting aside, it's a multiple choice exam. At least historically, it has been. Yeah, it's a multiple choice exam, and in general, what they have found is that a chimp will do better than a human, which basically means that random guessing is a more successful strategy than using your preconceived notions. So, what's the point here? Why are we doing this? I think that the Gapminder is interesting because it gives us a sense of the importance of having a sort of numerical sense of the world around us. And again, I've already mentioned that that's going to be one of the primary themes of this course. And so the Gapminder shows you that in the context of the world we live in. And our version tries to give you a sense of biology from the standpoint of the numbers. So Mark Kirschner is a very well-known cell biologist, very thoughtful guy at Harvard University. And Ron Milo, my book co-author on cell biology by the numbers, was a postdoc with him, and he once heard Mark make the remark that trying to understand molecular biology without numbers is like studying history without knowing geography, and I've always liked that. I think, for example, I've said this in the past, I think very much of Hannibal and crossing the Alps with his elephants and that sort of thing. It's a part of the real history. If you spend time in southwest France, you can really understand why there's a natural border basically between France and Spain. So as I said, this is really something that Ron Milo and Yinon Baron and I have had a lot of fun thinking about. And so this is the biological Gapminder, if you will. Before going on and giving you the exam itself, I just want to say a few more sort of philosophical things that shed light on the basis for this exam. So I will come back to this theme again in the course. It's one that I come back to whenever I have a chance because I feel it's in many ways so wrong-headed. There's tons of emphasis on surprise. And it's the sort of thing that reviewers will use as a reason to ding a particular paper. You hear it in conversations after talks or whatever, and I just don't. I'm strongly saying I don't support that as like a fundamental pillar upon which science should be built. Sure, that's fine for whether or not we're amused, but in terms of the business of making knowledge, I would say in some ways that science is about making unsurprised. The fact that we just a week ago saw the launch of the James Webb Telescope after 10 years. I assure you that nobody's interested in surprises, at least until the telescope is deployed. I think everyone absolutely expects there will be surprises, and I'm fine on that once the telescope is in use. And so this notion of surprise is kind of nuanced. There's two parts to it. There's the necessity of unsurprise, and all I really mean by that is, as I show on the right, one of my favorite all-time figures from any book or paper. This is from Jean Perrin, his book Atoms. It's, I think, the third to the last page, and gives you on the order of 15 different ways that Avogadro's number had been measured. And I think that in this day and age, in this moment, I'm giving this course online for the moment because we're living through a worldwide pandemic. I'm going to touch on this later, but I would say, the germ theory of disease, and I use the word theory intentionally. It's a big word. It's a good, strong word. It's not a word about casual understanding. You can't have a theory of why the food is bad at a restaurant. It's not a theory. A theory is a confirmed scientific hypothesis that's been tested. And so the germ theory of disease has been going for more than 100 years in the hands of Koch and Pasteur. We learned a lot about the nature of pathogens, and then in the early 1900s, late 1890s, early 1900s, we discovered viruses, and so we're living in a time where some large fraction of the population, I say large because if you go to a country like Iceland, I think you'll find almost nobody that would think of the current pandemic as a hoax, or that would be willing, themselves to sit there and say, yeah, I know best. I was recently on a flight on my way to Europe, and I was sitting behind a pilot. He was deadheading from Los Angeles back to Toronto, and I asked him what he had learned from the pandemic. And I'm not going to comment on that very much, but we got to talking, and he was weirded out by the lack of sort of belief or trust in knowledge. People trust their GPSs all the time without any question. And he was basically saying, why wouldn't people trust the germ theory of disease? And the comment that he and I had for each other is, no sensible person would imagine that they could go into the cockpit of his plane, and he flies a 777, and just sit there and fly it. No, it would never in a million years occur to somebody that actual expertise that a pilot has is somehow not really a very serious thing. Anyway, why am I making this big excursion? Because knowing things is hard, and we do know things. So on the left-hand side, I show you something I'm going to come back to again, which is a census of the various parts in the ATP synthase. And I just wanted to point out, the mass spectrometry results, if you look at the third row for item C, you'll see that the expected ratio and the measured ratio are, way, way, way off. And, this should give people pause. This should give people pause about, oh, well, it was already measured by person X. This is a kind of typical reviewer remark. Well, no, we need to measure things in more than one way. And I guess I should have asked Thomas' permission, but Thomas Lequi on the lower left, he had sent me a text at one point during the pandemic, just because he was talking about the weirdness of how misinformation propagates. Anyway, so beyond the data, we're going to talk about data, but fundamentally the reason I'm going through this biological numeracy exam is if I can't explain the basis of the data, then I don't really understand it. And so the spirit of that enterprise is going to be what we refer to as Fermi questions. Enrico Fermi was said to be able to get to a numerical answer to any question about physics, or probably most other things, in 15 minutes. And I think that that's a skill that we would want to emulate, to actually be able to, by using what we know about the laws of nature, by using our intuitions, by using our ability to make estimates, to be able to actually understand the scales that appear in things. And that's going to be a motif that runs through the whole course, which is, my diversion of developing intuition is really about what sets the scale of a given thing. So like, what sets the height of mountains? What sets the scale of synthetic nitrogen use? The claim is that we now feed twice as many people on planet Earth as we could in the absence of synthetic nitrogen fixation. What determines the stiffness of polymers, such as DNA or microtubules? What sets the time scale of evolution? I already talked about that in my I Wonder segment. And then the reason I show the Little Prince, you might remember that on a particularly sad day, he sat down and he watched something like 45 sunsets. It's a very small planet. It's rotating. The day is a very short period. And the reason I show it is just to note that on a very small planet where population sizes would be quite small, then the role of genetic drift becomes ever more important. And that's a very interesting thing, is the interplay between selection and drift, etc. So if I can't answer these kinds of what sets a scale questions, I really think I don't understand them. So with that as my background, here is the exam, and I'm just going to read each question and then we'll be done. And what I said is I would like for you to have a piece of paper, and you can use shorthand notation to yourself, but I would like you to make a go. So I'm giving you multiple choice. How big is the SARS-CoV-2 virion? And when I say big, in this case, I'm not talking about volume, I'm just talking about the linear dimensions. And if you like, think of it as a little ball. The question is, what's the diameter of that ball? What is the volume of a base pair? So I've already, I think in the I Wonder segment, mentioned this a little bit because I was amused and intrigued by the idea that engineering at the level of a few hydrogen bonds is sufficient to make the difference between somebody being sick and not. Both sickle cell anemia and cystic fibrosis are single, if I remember correctly, both single nucleotide mutation diseases. So what's the volume of the base pair? 1 nanometer cubed, 10 nanometers cubed, 100 nanometers cubed. What is the mass of an amino acid in Dalton units? And I remind you that Dalton is the mass of a hydrogen atom, or said differently, 1 gram per 1 gram per mole. So, so is the mass of an amino acid 10 Daltons, 100 Daltons, a few times 100 Daltons, or 1,000 Daltons? Which is bigger, protein or the mRNA that codes for it? So, we're used to thinking about blueprints being small in comparison with the buildings or the bridges or whatever structures it is that they represent. So here, I'm just asking, which is bigger? At cellular pH of 7, how many hydrogen ions are there per E. coli cell? Is it 100, 1,000, 10,000, 100,000, or a million? How many amino acids in a typical protein? 10, a few, 100, a few times 100, 1,000, a few times 1,000. See, I had a typo with my few times 0, 0. It's amusing. What's the volume of a bacterium? 10 to the 6 nanometers cubed, a micron cubed, 10 microns cubed. Maybe, in each of these cases, it'd be best if you were to justify your answer. How many genes in the human genome? 10 to the 4th, 10 to the 5th, 10 to the 6th. And I think I must have deleted it. How many genes in a typical bacterial genome as well? Excuse me, I'm having sort of some weird allergy this morning. What length book would DNA polymerase, including proofreading, copy without an error? Would it be the length of the Bible, the length of the Encyclopedia Britannica, or the entirety of English Wikipedia? How many carbon atoms are needed to make a new bacterium? Is it 10 million, a billion, 10 billion, 10 to the 20th, closing in on Avogadro's number? How many carbon atoms? How many sugar transporters are needed to make a bacterium? Is it 100, 1,000, 10,000, 100,000? Who is more powerful on a per kilogram basis, me or you or the sun? And by what factor? How long to exhaust the ATP metabolite pool in the absence of ATP synthase? In other words, if I just shut off all the ATP synthase at a given moment, then how long would it take to exhaust the ATP? What is the typical protein to mRNA ratio? And this is going to be independent of whether we're thinking about bacteria or yeast or human cell, roughly. Where is the most biomass harbored? Is it in arthropods? Is it bacteria? Is it viruses? Or is it plants? So that's my exam, and I'm looking forward to having you have a go at it, and we'll talk about the answers to those questions, at least the approximate answers, in the next vignette.